This is the real stuff of biochemistry. Biochemistry doesn't just happen in test tubes in the laboratory. It happens out here in nature. It's the driving force of life itself. Our countryside is really a huge biochemical factory, a machine for making, cycling and recycling the chemicals of life. Life on Earth is based on carbon, this black, uninspiring looking stuff. And before this twig was charred in a fire, it was the branch of a tree, and the carbon inside it was combined as a host of cellular organic molecules. And if you ignore the water slopping around inside cells, getting on for 50% of every living thing is carbon. But it's plants that have the carbon monopoly. There are plants everywhere, and trapped inside them are billions of tons of organic carbon. But where does the carbon come from in the first place? The answer, from thin air, from the tiny wisp, 0.04% of carbon dioxide in the atmosphere. In one of the oldest processes of life, this crucial raw material is constantly drawn into the carbon processing machine in plants. That machine turns the inorganic carbon in CO2 into organic material. And this stuff, all around me, is absolutely chock-a-block with it. And the recipe is so simple. It's bubbling away all around us, wherever and whenever there's sunshine. The sun, the power behind the biochemical factory. Once harnessed, the enormous energy of sunlight drives a crucial reaction, one that turns carbon dioxide and water into carbohydrate and oxygen. The recipe is called photosynthesis, and it's what makes England a green and pleasant land. And it's all a lovely green because plants are photosynthetically blind to green light. They absorb the red and blue part of the spectrum, but reflect back the green unused. It's only with light that we get life. Young plants struggle upwards, hungrily soaking up the sun's rays, each leaf angled to follow the sun, grabbing as much energy as it can. That energy is captured in the green chlorophyll in the green chloroplasts within the leaf cells. It powers the breakdown of water molecules, throwing out oxygen, and then, with what's left, reducing carbon dioxide to organic compounds stored in the leaf. The water for the reaction passes in from the soil through tiny root hairs into the cells of the root. Then, from the darkness of the soil, the water passes through special tubes within the stem, on and up to the sunlit canopy above, and to its rendezvous with CO2 in the machinery of the chloroplast. And carbon dioxide, it simply enters the leaf through a myriad of tiny pores, stomata, that pierce the blade of every leaf. Photosynthesis is all very well for plants, but what about us animals? We can't make organic materials from carbon dioxide, so what do? Well, one way or another, we eat plants. And for many animals, just being a plant eater is enough. But some animals eat the animals that eat plants, leaving behind the evidence of the food chain from plant to animal to animal. Others eke out a grisly existence, slowly but surely cleaning up the debris and detritus of death itself. And some, exploiting the organic material left unused by others, revel in the fecal pleasures of life. Whether living things get their energy first, second, or even third hand, everything is ultimately powered by the sun. And by powering the synthesis of the compounds of life, it gives us both the building blocks of our very selves and the fuel to make it all work. What sort of work? The precise swing of the blacksmith's hammer, a coordination of eye and arm, controlled by a stream of signals, work done in the transport of ions in the nerve cells. The kinetic energy in the falling hammer, working, shaping and reshaping the iron rod, powered by the muscles in the arm. The muscles themselves, protein blocks that must be built up from raw materials. And always heat, the inescapable byproduct of anything metabolic. All elements of life demanding energy expenditure and an input of fuel. That fuel is sugar. We get our sugars from our food, of course. But how do we unlock the power that's inside those sugars? Well, glucose is effectively a fuel, as much a fuel as the coal that we're burning on the furnace. In the flame, we've got the reverse of photosynthesis. The sun's energy that was locked away millions of years ago is released when we burn it. And by burn it, we really mean oxidation. And for that, we need lots of oxygen. Oxidation, the fire of life. Through oxidation, we make the chemical energy of sugar available, simply converting it from one form to another. Movement, biosynthesis, active transport, and heat. But how much energy do we get from our sugar fuels when we stoke the fires of life? The amount of energy, even in this tiny little sugar lump, is really impressive. In fact, there's probably enough energy in the sugar in my tea to keep me going for the best part of half an hour, or even a bit longer. But it's all chemically locked up. And to get it out, I need oxygen. This time, I'm using liquid oxygen. And then burn it. Well, I'm pleased to say that that all-at-once fireball release of energy doesn't happen inside us. Instead, the energy, the chemical cell energy of the glucose molecule, is released in a series of little steps. Gradually, that is, in the various steps and stages of catabolic pathways. It's rather like the flow of water through a series of canal locks. Nothing much happens for a while in energetic terms. One chemical is gradually converted into another inside the cell with no major change in energy level. And then there's a sudden, significant release of energy. Metabolites rush through from one reaction to the next, only to be followed by another bit of flat, so to speak, before moving on to the next reaction in the pathway. Whatever the analogy, a flight of locks or a flight of steps, the overall idea is the same. One metabolite is converted to another with a big difference in energy levels between the two. That difference is available energy, and when released in safe, manageable stages, it can be captured and used by an organism. But how is it caught and used? There's life even in the Grand Union Canal. I've certainly got some waterweed, and with a bit of luck, I've got what I'm really after. Yes, there are lots of them, tiny dancing specks of life. Daphnia. Water fleas, if you like. Let's go inside and bully them a bit. What I've got here is an oxygen electrode. It's full to the brim with water, fully saturated with oxygen. Down the bottom, there's a sensor that measures oxygen concentration and a wire taking the signal to the chart recorder. Well, I'll just switch on, pen down, and yes, straight away I'm getting a nice trace, straight line, maximum oxygen concentration. So what I'll do is add some of these beasties. Yeah, I've got some. Put them in there. I can hear things happening straight away, so we should be all right. Put the top on to keep out atmospheric oxygen. And, oh gosh, yes, it's really going down straight away, a lovely line. In fact, it's more or less plummeting down. And that means that oxygen is being used like mad. It's respiring away like anything. And that, of course, is oxidation. Not just in the Daphnia, but in each and every cell inside them. The question is, where precisely in the cell is it all going on? Where are those biochemical fires? Well, to answer that, we've got to divide the cell up. Well, I'm going to leave the Daphnia alone. I think they've done enough. And we'll concentrate on the waterweed. It's okay to use waterweed because, as well as doing all that photosynthesis stuff, they respire just as much as animals do. But to avoid any complication and confusion, it'd be better to concentrate on roots. These actually haven't got much, so I think we'll use this bigger, juicier-looking beast here. There aren't any chloroplasts in the roots, so that should be absolutely fine. And then, to smash the cells up, we need to use a homogenizer.
And it's done like this, even in the best biochemical circles.
And what I end up with is plant root soup, or soup of plant root if you prefer. And to separate out the components, you need to give it a spin in a centrifuge. And I've got here the results of a 10-minute spin. And if I get the tube out, at the top there's a cytosol water mixture, and at the bottom the heavier, denser components like nuclei, plant cell walls, and mitochondria. And we can test that lot in the oxygen electrode assembly. When this was intact, it used oxygen. And if we haven't damaged it too much in everything we've done to it, there should be something in here somewhere that will do the actual oxidizing. The question is, what? Well, got a fresh, new oxygen electrode assembly. Clean up a bit. Switch in the new oxygen electrode. That's fine, I'm getting a nice starting line there. So what I'll do is add some of the cytosol and see what happens. Okay, off on. Well, I can hear something. Well, nothing's happening. It's flat. Well, that must mean that there's no oxidizing stuff here in the cytosol, which means we must hope that it's in the sediment. Let's try. Let me get rid of some of the cytosol. There. And resuspend the residue and add some of that to the electrode. There we are. Now then. Well, that's better. It's going. It isn't roaring down, but it's going. But there's certainly oxidation there. The question is, where? Well, we could separate the components out further, if we wanted to, but we can get quite a long way by the process of elimination. It isn't likely to be the nuclei. After all, they're mainly DNA and the cell's genetic material. It almost certainly isn't cellulose cell walls. They're dead and inert. In fact, that only leaves mitochondria, and they really are our prime candidate as the site of oxidation. We can try that. We can test that idea out by injecting into the cell some pyruvic acid, which is, of course, the end product of glycolysis, a super generator of NADH2, absolutely marvellous food for mitochondria. And wow, they're really going. In fact, I'm going to add something else quick while I've got a chance. Right, in. And it's absolutely flat. It's absolutely stopped it stone dead. And what I've added is cyanide ion, and that totally, irreversibly inhibits cytochrome A3 in the electron transport chain. And that's why cyanide is the poisoner's favourite tipple. It rapidly and irreversibly poisons mitochondria and kills you stone dead. But assuming you avoid a cyanide tipple at the local pub, the oxidation of sugars, the fires of life, burn constantly in the mitochondria. And of course it's not really lots of little fires, although heat certainly is given off. Instead, life uses a special sort of energy currency called ATP. That ATP currency is used throughout the body. Anywhere that energy is needed, wielding a blacksmith's hammer or lifting this glass, the local cells get their mitochondria to make some ATP. The question is, how do they do it? This is NADH2. It's made in the glycolytic pathway, the link reaction, the TCA cycle, and it, together with its biochemical twin, FADH2, is oxidized in the electron transport chain in the billions of mitochondria throughout my body. The overall reaction is simple. Look. NADH2 reacts with oxygen to give me my NAD back again, and water. And of course we get some heat and our 3 ATPs. But of course, as always, the reaction happens in lots of little stages. In fact, NADH2 is converted to NAD, and what does it is coenzyme Q itself becoming converted to QH2. And the energy of that redox reaction is captured in the form of ATP. And then, abracadabra, what happens next is that oxidized cytochrome B is converted to reduced cytochrome B using the H2s of QH2. And then, again and again and again, I've got 3 stages compressed into one there, and the various oxidized cytochromes are converted to reduced cytochromes, and we get another ATP. And then, finally, bingo, oxygen does its stuff, and we get our last ATP. But of course, if we'd started with FADH2, what we'd have done is completely block out this site and ended up with just 2 ATPs overall. So in the whole process, a pair of hydrogens passes right the way down the chain in a sort of biochemical dance. Look, beginning again, an enzyme takes a pair of hydrogen atoms from an organic compound, gives them to NAD, and the dance begins. Keep a close eye on that pair of roses, hydrogens, passing from dancer to dancer down the transport chain. The white hankies twirl in as ADP and then out again, converted to ATP. And of course, those hydrogens are immediately followed down the chain by another pair. The stage for this dance is the inner membrane of the mitochondrion, and it goes on in parallel in every cell in the body. It is, of course, a fantastically complicated series of reactions, but the essence is a flow of hydrogens and of electrons from NADH2 down the chain in that series of little steps. And the flow goes on, carrier to carrier, the energy of the progressive oxidations raising low energy ADP to high energy ATP, 3 times for each pass down the chain. Then, at last, the roses reach the ultimate hydrogen acceptor, the oxygen of air itself. And the result is water. So the hydrogens of NADH2 and FADH2 eventually meet the oxygen we breathe deep inside the mitochondria. The result, an outpouring of ATP driving the machinery of life.
That dance happens about 1 million times every second in every cell of my body. A grand cycling of ATP to ADP and back again. And that cycle is matched by others. Carbon in carbon dioxide is converted to organic carbon and back again. The oxygen in water goes to molecular oxygen and back again. So cycles of elements and compounds driven by the energy of the sun through life to the inevitable product, heat. The fires of life in a nutshell.